Good morning, everyone. My name is Bibiana Jaramillo, and today I will present three experiments that we conduct about the effects of greater levels of phytase on energy, amino acids, calcium, and phosphorus digestibility in soybean meal fed to growing pigs. In diets for pigs based on cereal grains and oilseed meals, most phosphorus is unavailable because it is bound to phytate, resulting in low digestibility of phosphorus. Phytate is negative charged, allowing interaction with some minerals such calcium. Phytate may also form binary complex with charged amino acids and protein, especially at low pH, and can also participate in ternary complex included starch when minerals are present and pH is higher. However, use of exogenous microbial phytase in diets for pigs results in hydrolysis of the ester bonds between phosphorus and phytate, which results in increased digestibility of phosphorus and calcium, with a subsequent increase in absorption of both minerals and maybe increase the availability of other nutrients. On the other hand, soybean meal is the most widely used protein ingredient in pig diets, and in corn-based formulation, soybean meal is the main source of an indispensable amino acid. Soybean meal also contributes substantially to the energy in diets, and concentration of phosphorus for soybean meal is higher than in the corn. Nutritional matrix values for precise diet formulation using soybean meal and superdosing of phytase are needed. That's why three experiments were conducted to test the hypothesis that the apparent total tract digestibility, or ATTD, of energy and standardized total tract digestibility, or STTD, of calcium and apparent and standardized ileal digestibility of amino acid in soybean meal increase as more phytase is added to the diet. For these experiments, three sources of soybean meal were procured from South Dakota, Illinois, and Indiana, and then were labeled as a soybean meal 1, 2, and 3. The three sources were analyzed for gross energy, crude protein, amino acids, fat, fiber, free sugar, anti-nutritional phytate, trypsin inhibitors, and minerals. As I summarized, no difference between gross energy and crude protein were found for the three sources of soybean meal. However, calcium was higher for soybean meal 03. In the first experiments, effects if greater levels of phytase on digestibility of energy and concentration of digestible and metabolizable energy in soybean meal were measured. Each source of soybean meal was included in 5 corn soybean meal diets containing 0, 500, 1,000, 2,000, or 4,000 of phytase per kilo of diet. And a corn-based diet was also formulated. Therefore, a total of 16 diets were used. 128 pigs with a body weight of 15 kilos were housed individually in metabolism crates that were equipped with a screen floor for total fecal collection and urine pan that were installed below the slated floor. Pigs were allotted to the 16 diets using a randomized complete block design with 4 blocks with 32 pigs, 2 pigs per diet, and a total of 8 replicates per diet. For collection and sampling, the initial 5 days were the adaptation period to the diet, which was followed by 4 days of collection. In short, on day 6, a color marker was added to the morning meal to mark the beginning of the fecal collection and the stop marker in the morning of day 10. At the conclusion of the experiment, fecal samples were dried and then analyzed for gross energy. Urine samples were towed, filtered, and mixed, and a subsample was put in a freeze dryer to then be analyzed for gross energy. The statistic analysis include mixed procedures in SAS. The initial model includes soybean meal source, phytase, and the interaction between both. However, no interaction between soybean meal source and phytase were observed, and the final model, therefore, containing only levels of phytase. Moving to the results, the observation that there were no interactions between soybean meal and phytase indicates that all sources had a similar response to the phytase, which is in agreement with previous data. On the x-axis, the different levels of phytase supplementation are presented, ranging from 0 to 4,000. On the y-axis, fecal output is expressed as a grams per day. As phytase inclusion increased, fecal output decreased. Quadratic indicated an improvement nutrient utilization and reduced excretion. Therefore, the ATTD of dry matter in diets increased with increased phytase inclusion. The ATTD of gross energy also increased as phytase inclusion increased, indicated that some energy containing nutrients in soybean meal were bound to the phytate, and those effects reduce the digestibility of energy. But phytase reduce that negative effect. The digestible energy and metabolizable energy of diets increase when increased levels of phytase were fed. This observation, where the digestibility of energy increase with phytase, indicates that some energy containing nutrients in soybean meal were bound to the phytate again, and the inclusion of phytase reduced that negative effect. As a conclusions, not interactions between soybean meal and source of phytase. There were no difference among soybean meal source, and higher levels of phytase can increase gross energy and digestible energy. Phytase may also increase digestible energy in soybean meal by 40 to 50 kilocalories per kilo. Moving to the next experiment, increasing levels of phytase in corn soybean meal diets increases the STTD of calcium in soybean meal when fed to growing pigs. 3 corn soybean meal diets were formulated using each of the 3 sources of soybean meal. 12 additional diets were formulated by supplementing each basal diet with the levels of phytase before described. However, because diets were based on corn soybean meal, we had a deficiency of calcium. In this case, we had to use limestone in all diets to get enough calcium. That means that the digestibility we measure in these diets was a combination of the digestibility of soybean meal and limestone. That's why 5 more diets based on potato protein and limestone were formulated to get the digestibility of limestone in each of the levels of phytase. And then we could use those values to correct the values in corn, soybean meal diets and calculate the digestibility of calcium just in the soybean meal by difference. For experimental diets, 160 pigs with a body weight of 15 kilos were allotted to 20 diets using a randomized complete block design with 4 blocks of 40 pigs, 2 pigs per diet in each block for a total of 8 replicate pigs per diet. For collection and sampling, we use the same marker-to-marker collection methodology, but in this case, we just took fecal samples and they were dry and analyzed for dry matter, ash, calcium, and phosphorus. Moving to the results, apparent total tract digestibility of calcium and standardized digestibility of calcium in soybean meal was increased as phytase levels increased. The observation that microbial phytase increased the digestibility of calcium in soybean meal supports the hypothesis that between inositol and phosphorus can be hydroxylated by phytase and thereby also release the calcium that was bound to the phosphorus in the phytase complex. Moving to the conclusions, the release of calcium from soybean meal increased as phytase increase. Higher levels of phytase can increase ATTD of dry matter. Greater levels of phytase increase STTD of calcium in soybean meal. Moving to the third experiment, the effects of greater levels of phytase on standardized ileal digestibility of amino acid in soybean meal fed to growing pigs. We formulate a total of 16 diets. Each source of soybean meal was included in 5 cornstarch soybean meal diets containing 0, 500, 1, 2, and 4,000 units of phytase. All diets, except the nitrogen-free diets, were formulated to meet nutrients requirements for 25 to 50 kilograms pigs, according to NRC 2012. The soybean meal included in each diet was the sole source of amino acids. All diets also contained 0.4% of chromic oxide as an digestible marker. For experimental design, 16 growing barrows were equipped with a T cannula in the distant ileum. After 6 or 7 days of recovery, the pigs were weighed and allotted to one of 16 diets in a 16 by 8 Uden square design with 16 pigs, 8 periods, and 1 pig per treatment in each period. The initial 5 days of each period were considered an adaptation period to the diet, and then ileal digesta were collected for 9 hours on day 6 and 7 of each period using. Moving to the results, the standardized ileal digestibility of crude protein is increased by increased levels of phytase in diets. This is in agreement with previous data where microbial phytase increased the digestibility of amino acids in pigs. The standardized ileal digestibility of indispensable and dispensable amino acids were increased as microbial phytase was increased in the diets. Here, the digestibility of some indispensable amino acids such lysine, methionine, threonine, tryptophan, and valine. The observation that digestibility of all amino acids increased with phytase indicates that phytase improved utilization of protein-bound amino acids in the diet to the phytate, and the response was not only a consequence of reduce of endogenous losses. As a conclusion, phytase superdoses increase digestibility of all amino acids regardless of soybean meal source. Soybean meal source had only minor effects on standardized ileal digestibility of amino acids. The result that the response to phytate was consistent among the 3 sources of soybean meal has practical implications because it indicates that matrix values for digestible amino acid released by phytase may be used for soybean meal regardless of the sources used. However, because only soybean meal that was procured from the United States was used in the present work, it is not known if the standardized ileal digestibility of amino acids in soybean meal from other origins also is increased by microbial phytase. But future research should be directly determine the impact of phytase on digestibility of amino acids in soybean meal procured from outside the United States. Thank you to the Hans Stein Monogastric Nutrition Laboratory and our sponsor, United Soybean Board.